Biologists have long debated exactly where the line is drawn between a living organism, a dead cell, and a piece of engineered machinery. Synthetic biology is now moving beyond simply editing existing DNA. A new frontier in the field involves writing entire genomes from scratch to command exactly how a cell functions. But there's a massive biological obstacle standing in the way of these genome transfers. Living cells have strict evolutionary defense mechanisms designed to fight off foreign genetic takeovers. They employ a process called homologous recombination. When faced with a massive engineered genome, the host cell actively snips out and steals only the tiny, useful fragments, like a specific gene for antibiotic resistance. It then completely rejects the rest of the invading sequence. This fragment stealing tactic creates incredibly frustrating false positives. A cell lights up, surviving an antibiotic test, tricking researchers into believing a complete synthetic transplant was successful when it actually failed. That innate self-preservation makes the living cell a terrible host. To build a fully compliant organism, researchers realized they had to completely neutralize the original life first. Researchers at the J. Craig Venter Institute in California have spent over a decade pursuing the creation of a fully synthetic cell. They realized that as long as they tried to negotiate with a living, active host, they were going to lose. The host's biology would always fight back. They needed a completely blank slate. The only way to bypass those natural biological defenses and eliminate the false positives was to initiate a controlled, functional cellular death. To test this, the researchers started with a population of a specific bacterium called Mycoplasma capriculum. They exposed this bacterial population to mitomycin C, a powerful chemotherapy drug known for causing severe DNA damage. The drug performed a highly specific demolition. It totally dissolved the cell's native genetic instructions, but it left the outer physical membrane completely intact. This induced state left the cells functionally dead. Without their native DNA, they were completely incapable of replicating and incapable of performing homologous recombination. This chemical deactivation perfectly separated the physical hardware of the cell from its genetic software. The host was now just an empty receptacle. With the host cleared out, the researchers introduced a fully synthesized engineered genome from a closely related species, Mycoplasma mycoides, into these dormant cellular shells. The new synthetic genetic material physically merges with the deactivated empty host body. Against the odds, a small fraction of these functionally dead cells successfully absorbed the new genome, rebooted their systems, and resumed biological function. The entity functioning in the petri dish no longer possesses its original DNA, but it also doesn't possess the physical body of the engineered donor species. This resurrected chimera is what the JCVI team calls a zombie cell, a biological host running on a completely artificial operating system. This zombie cell method definitively solves the false positive problem that has plagued geneticists for years. Dead cells simply cannot steal stray DNA fragments. Now that the technique works, researchers are preparing to move away from testing on highly specific niche bacteria and start applying this method to robust, widely studied species. The primary future candidate for this blank slate genome swapping technique is the laboratory staple E. coli, better known as E. coli. Standardizing this resurrection technique transforms custom genome transplantation from a rare, unpredictable anomaly into a scalable, universal tool for laboratories worldwide. On an industrial scale, these fully reprogrammable cells can act as obedient microscopic factories. They can manufacture complex pharmaceuticals, mass-produce customized vaccines, without native biology interfering. Environmentally, these uncompromised cellular shells could be programmed to efficiently process waste or produce sustainable, next-generation biofuels, multiplying exactly as designed. By figuring out exactly how to induce and manage cellular death, researchers have bypassed the natural defenses that keep species separate. We now have a reliable method to author entirely new forms of life.